Hello, everybody. I'm Congressman Jimmy Panetta, and I represent the Central Coast of California, the place that I've always called home, the place where my Italian immigrant grandfather decided to call home when he settled here, and the place that I unabashedly and proudly call the most beautiful congressional district in the nation. I introduced myself that way to my congressional colleagues, and they didn't take too kindly to that statement, to which I responded, well, I guess you've never been to the Central Coast of California. Now, many of you have been here, so many of you appreciate the beauty here because many of you have worked so hard to protect the oceans that are here, not just on the Central Coast, but coast to coast of our great nation. So it's a real honor that I get to welcome all of you to this session and thank you, not just for your work in the past, but also your work today as you discuss, as you listen, and as you learn about innovative approaches for restoration and monitoring of our oceans. Now, on the Central Coast, in our front yard, we have the National Monterey Bay National Marine Sanctuary, we have the California Coastal Monument, we have the Elkhorn Slough National Reserve, and numerous research labs that line the Monterey Bay. And in our backyard, we have the Silicon Valley. That setup allows us to have an ideal living laboratory and test bed for applying innovative technologies to marine conservation, from managing fisheries to detecting changes in critical habitats like our kelp forest and unlocking the past biodiversity so that we can look to the future. Now, being from here with its beauty and innovation, it really does drive a portion of my work in Washington, D.C. That's why I introduced the National Oceanic Partnership Program Reauthorization Act to pave the way for more private funding to bolster federal programs. It's also why I introduced an amendment to the Coastal and Great Lakes Communities Enhancement Act that encourages collaborations and partnerships between institutions of higher learning and federal agencies by expanding data sets used for coastal management. Our oceans and our technology is what drives the work of many of our panelists today, which is why we anticipate their discussion on how we can take advantage of technology to bolster the protection and endurance of our oceans. And it's their work that drives many of you to be a part of this discussion today so that all of us together can fight for our oceans tomorrow. Thank you. Enjoy the panel.
Hey everyone, this is Congressman Brian Mast. I hope you're all doing well and I hope you're all staying safe. Now, even though we can't get together in person right now, I wanted to take just a minute and say welcome to this year's virtual Capitol Hill Ocean Week. The community I represent in Southeast Florida is home to some of the most biodiverse ecosystems in the entire world, including the St. Lucie Estuary, which I live on, the Indian River Lagoon, the Loxahatchee River. Our oceans and waterways are truly a precious resource that myself and my family love very personally. That's why since coming to Congress, I've made it my primary mission to protect Florida's waters, do everything in my power to promote healthier, more biodiverse ecosystems that allow our communities' fishing and tourism economy to thrive. Now, I continue to be inspired by the new technology being developed each and every year that allows us to find new and innovative ways to promote our ecosystems, protect our waters. It's in that I just want to say to you all again, to all of you, thank you for joining us in this year's virtual conference, and I wish you all the best. Y'all take care.
Thank you for joining us today and for being flexible as we adapt to dealing with COVID-19. I hope you are all well and your families are safe. Special thank you to the Sanctuaries Foundation for hosting this panel and for their staff for helping us adapt to this virtual world. I'm Josie Quintrell, and I am the director of the IOOS Association. We're a nationwide nonprofit dedicated to delivering science, data, and information on our oceans, coast, and Great Lakes. We do this through supporting the Integrated Ocean Observing System, IOOS. It's the nation's coastal and ocean observing system that links together a network of 11 regional observing systems with 17 federal agencies to provide an integrated approach between the federal and non-federal and industry partners. We're stakeholder-driven and science-based. We like to say we measure once and use multiple times. So our data is used to support safe and efficient maritime commerce and to prepare and plan and respond to coastal hazards. Understanding and tracking the rich biodiversity of our nation's oceans is a priority for IOOS. IOOS is taking an active leading role in networks such as the Marine Biodiversity Observing Network, the Animal Telemetry Network, and we're constantly trying to figure out how we can improve our ecosystem monitoring. And that's why I'm really happy and thrilled to be moderating this panel today on innovative approaches to monitoring and conservation. We have a diverse panel for you today that will highlight the role the U.S. is taking in marine biodiversity from the intertidal level to the global ocean. We're taking a broad approach to thinking about innovation and will not only explore exciting new technologies such as eDNA and machine learning, but also on how low-tech approaches can be applied in innovative ways, and explore innovations in partnerships and citizen sciences, all with the goal of trying to provide a glimpse at how new activities are making monitoring more efficient and more effective, and hopefully more cost effective. So I'm going to provide a short intro to each of our speakers today. You can find their full bio on the Cattiau website, so I encourage you to go and look at that. So we're going to start off the conversation with Dr. Frank Muller-Karger, who is a professor at the University of South Florida. He is also a lead principal investigator on the Marine Biological Observing Network. And so I'm going to turn it over to Frank.
Very good. Thank you, Josie, and hello, everybody. I want to emphasize something that Josie alluded to, and that is that one of the things that we need to do to monitor and to capture data is transform that into information. And one of the limitations that we have, not just in any locality, but around the world, is the amount of information about biological diversity that we can collect or that we have collected historically. And so this is something that there's very few observations, and they're hard to come by, they're hard to process, to put into common databases. And so one of the objectives of the Marine Biodiversity Observation Network is to get that information flowing again. And so we, in the US, we're sponsored by NASA, NOAA, BOEM, ONR, and IOOS under the National Ocean Partnership Program. And so I'm thankful for them for spearheading this. So I'm going to show you some of the technologies that people are using right now to try to accelerate the collection of information. And for example, I'm going to try to go up from very small things to larger things. Here, for example, there's a device that can be automated or moored. And that's an imaging flow cytobot, bot, and takes pictures and also gives you information about phytoplankton taxonomy. And that's associated with artificial intelligence, for example, that allows you to classify them and identify them automatically. And then that would go into an online database. There's also similar things for a larger, slightly larger size class that is called a UVP, Underwater Vision Profiler. And that gives you information about zooplankton. And so you can see on the right-hand side bar graphs and some plots on the information that it gives you. So very, very important. If you combine these types of technologies, you start getting a picture of the lower trophic level, so the organisms that feed everything else. Then there is a very large number of technologies that you can attach to animals and have them move around with these devices and track them, track their position, track their behavior, but also collect measurements about the environment. And that is something that the Animal Telemetry Network is trying to collect and make openly available. And so we're trying to connect the marine biodiversity observation. For example, if you have many measurements of this class, you try to aggregate them in, for example, in a GIS system. You could try to understand where you find hotspots of biodiversity, along with how the environment is changing. Another technology that's very exciting is what we call environmental DNA. That's part of the omics family. Here you collect a water sample and then you try to process it with a number of primers. These primers are basically a way to identify specific groups of organisms. So there's primers for bacteria and phytoplankton. There's other primers for invertebrates and others for vertebrates. And so if you collect all of these information with these different primers, you can put it together and try to say who was in what water parcel at any one point. And then there's acoustics, and acoustics is just a very, very broad area of research, and also in applications. You have active and you have passive acoustics. This graph shows you some of the things that you can identify with active acoustics, so a pinger or a device that emits sounds, and you can collect the signals back, and depending on the frequency, you can see different things. Then we can try to combine that with a number of sensors that we fly either in aircraft or in space. And especially in space, we can collect data for around the world every few days. And if you combine data from one sensor like ocean color with something like temperature or winds or waves or sea surface height, you can say a lot about the environment. And we're trying to combine these different data sets into one group of products that we call seascapes. And that's a biogeographic representation that is changing with time, and then you can combine it with the information that we had before. Ultimately, you need to combine all of this. So data systems are really important. You need to work with partners that provide information. You can put that information in an open system where you can aggregate it and then develop information or knowledge that you can deliver. For example, we work very closely with the National Marine Sanctuaries and try to update their condition reports. But ultimately, you can try to provide information to bigger international efforts like the Sustainable Development Goals. So one example of this may be, for example, in the Florida Keys, that you have here a picture on your left-hand side. They're trying to restore reefs. And so one thing that you would like to do is understand how biodiversity is changing as the reef matures, if the restoration effort is successful. So if we combine these different technologies, we can then provide information to the sanctuary and say, yeah, in fact, you have other organisms, not just the reef growing, but it is leading to a change in biodiversity that is useful and can be used by people. So anyway, that's it, Josie, thank you.
Thank you, Frank. That was great, and a great introduction to some of the technologies that are being used today. So now we're going to go out to the California coast with Dr. Jen Caselle, who is a professor at the University of California at Santa Barbara. And she's going to help us understand how some of these tools and techniques and other ones are being used in MPA monitoring. Jen.
Thank you, Josie. Great. Okay, thank you. Yes, so as Josie mentioned, I'm a research professor at the University of California, Santa Barbara, where I study nearshore coastal ecosystems like kelp forests and coral reefs. And my research is particularly interested in how these systems are affected by humans and environmental change, and how we can protect and restore these systems into the future. So one of my main projects for quite a few years now, actually for several decades, has been working with the state of California and a huge number of partners on implementing and monitoring the California Network of Marine Protected Areas. And this map shows those. So beginning in 1999, following the passage of legislation, which was called the Marine Life Protection Act, the state undertook a lengthy and highly participatory process to implement a network of MPAs across the state. And it took over 10 years. It involved a huge array of academic and agency scientists and policymakers, managers, and stakeholders from all over the state, sitting around tables and figuring this all out. And by 2011, we had a network of 132 marine protected areas in the nearshore waters out to the state limits along this coastline that is about 840 miles in length. And these MPAs, they span all, as I said, all of the coastal habitats, from the shallowest nearshore environments, the rocky intertidal and so forth, right on down to the deepest canyons and some of the seamounts. And so it's been a particular challenge for the state to think about as we move from implementation into the monitoring phase and adaptive management of how to do this. So I'll take you through some of the habitats that we're currently monitoring, as I said, from the very near shore, the rocky intertidal, the sandy beaches. These MPAs include all of these habitats. Moving a little bit deeper, we monitor the nearshore rock habitats, which in California are mainly kelp forests, rocky reefs. And although the tools that we use are not all that high tech, as we move into deeper waters, we employ cameras, drop cameras, ROVs, submarines, and the tools get a little bit more technological. So this was and I particularly want to mention that although the monitoring, it does include academic and agency scientists, many of the projects are citizen science projects. And this is a picture of one of my projects, utilizing volunteer recreational fishermen to monitor the marine protected areas inside and out using hook and line tools. So the Marine Life Protection Act, it was an unfunded mandate passed by the voters of California, and it required several things to be effective: funding and resources, serious mobilization of the scientific community. There was a lead agency, the California Department of Fish and Wildlife, but it's required coordination with lots of other agencies. And really importantly, a lot of outreach and collaboration with communities coming from disparate parts of the state and different kinds of communities. And I think the key to success of the California Marine Protected Area Network was definitely partnerships. They were and they continue to be key partnerships across all aspects, and from implementation into now the monitoring phase. In fact, these partnerships have been formalized by the state in a document that I'm showing you here, the California Collaborative Approach. And a lot of thinking has gone into how these partnerships can be successful. So now that we're monitoring the MPAs, we've still maintained a very, very high level of coordination. We have big projects that focus on each of those habitat types that I showed you, as well as some additional ones that I didn't show you. The teams are multidisciplinary and they involve people up and down the state. And then each of those habitat-specific projects works together to synthesize the data sets and to make sure that we're addressing some of the key management questions that the state of California is posing in similar ways or even in identical ways. So I'm going to leave it here and say that during this panel, I'd be happy to chat more about the specific ways that we're accomplishing the challenge of monitoring marine environments across 800 miles of coastline and at least 6 really different ecosystems. And also ways in which we're maintaining these really strong partnerships, both to fund and to do this monitoring and adaptive management. So thanks, Josie. I'll unshare my screen now.
Yeah, thanks, Jen. That was a really great overview. And obviously there's a lot to explore there in terms of your use of people and technology, and then also coordination. So going to move along the coast of California, and I'm going to hear from Dr. Jim Berry now, who is a senior scientist at the Monterey Bay Research Aquarium, MBARI. He's going to talk about his work on deep corals and how that monitoring is going. So Jim, I'm going to turn it over to you.
I'm delighted to be involved in this panel. It's something that's kind of close to my heart, trying to understand biodiversity, how we monitor ecosystems, whether it's intertidal all the way down to the deep sea. And I'm going to focus on the deep sea today and try to give you a quick overview of some of the things that are going on, some of the technologies that are required that MBARI is involved with, and also collaborating with the National Marine Sanctuaries in particular. And MBARI, just as a really quick bit of background, was started about 3 years after the Monterey Bay Aquarium was begun, and David Packard started MBARI with the idea that he would bring together scientists and engineers so that they could make progress in key areas in ocean science that are limited by technology. So developing new methods, new sensors, new techniques, new devices that can open up areas of exploration, discovery, and advancement and understanding so we can make informed decisions about the future of the oceans. So with that brief introduction, how do we work in the deep sea? There are some specific challenges, and the primary challenge is access. How do we get down there? How do we make measurements collect samples, explore areas in a way that we would if we were walking across a terrestrial landscape. And so we need ships like the Western Flyer that we have as our flagship, advanced underwater robotics like the remotely operated vehicles that we run. This is one of them, the Doc Ricketts, that dives to 4,000 meters. We have mapping systems like this AUV mapper, which can provide very high resolution maps of the seabed. And that maps are critical to undersea exploration, because these assets that we use to explore, the ROVs in particular, they're tactical, and we need to know where should we put those. Having high resolution maps allow us to target specific locations that might have some feature of interest, whether it's a hydrothermal chimney or a rocky ridge or whatever the target might be. So maps are very important. We also have more advanced long-range AUVs that can stay out for up to weeks and carry along with them some complex payloads like this sampler that is designed to take samples based on the exact conditions under which you want to take that sample. Where is the maximum in chlorophyll in the water column, or a particular temperature horizon? And then those water samples might be analyzed aboard or brought back to the lab, preserved and analyzed for eDNA, other molecular studies, or whatever else might be going on. So we have fleets of these that can go out together, each with different suites of sensors that can provide a comprehensive picture of what's going on in the environment, and then have a sampler go in and target those locations for sampling on these fine scale oceanographic features, or near the bottom even in other cases. Others, like on the lower right here, the seafloor rover that Ken Smith operates, is able to have deployments that last a year. It goes to sea for years at a time, well, coming up once a year to have its batteries replenished and checked out. And it can take metabolic measurements of the seabed, imagery, scanning locally, so giving you a much more high resolution in time picture of what's happening on seabed communities. And we also deploy time-lapse cameras in different environments, and I'll talk about those a little bit today briefly, and with coral communities like you see in the background behind me with this beautiful bamboo coral. So moving on to deep coral and sponge communities, these are systems that are oases of life in the deep sea, and they're these beautiful coral gardens that we have on many of the seamounts and rocky ridges along our coastline that differ from shallow water corals. Unlike shallow water coral reefs in the tropics that have microscopic algae as symbiotic species in their tissues, deep sea corals don't have symbiotic algae, and they depend upon the rain of organic debris from the surface water. So they're capturing particles like small sea anemones might do, for example. They're highly diverse systems. They're important for fisheries. They became notable really in the early 1980s, when you may recall the orange roughy fishery off Australia was booming, and what they were doing is trawling the tops of seamounts and destroying these coral communities. And this coral, I think it's the same coral I'm showing you behind me on the lower right here, they're highly vulnerable to trawl damage. They grow very slowly. The coral that you see behind me is easily 500 years old, if not much older than that. They take a long time to mature, and likewise some of these sponge communities may take a long time. So when they are damaged by any sort of human activities like trawling or even trap fisheries, they might be damaged for decades to centuries, and we want to figure out how to cope with that. NOAA has recognized deep sea coral and sponge communities as a very high priority. There are a couple of recent reports on the status or the priorities for research in these communities. We are right now in the midst of NOAA's West Coast Deep Sea Coral Initiative, in which we are collaborating with NOAA to work on some areas along our coast near Monterey Bay, and in particular with the Monterey Bay National Marine Sanctuary. And NOAA in general is surveying areas far outside and much broader along the US West Coast. So we in Monterey, and with the Aquarium, with NOAA in Monterey, are we have a deep sea coral initiative, or deep sea coral observatory, what we call it, where we've placed a variety of oceanographic sensors that tell us something about flow on small scales or larger scales near the corals, the flux of carbon from surface water, so that we can understand what sort of levels of food are entering the system over the year. We sample water near corals and sponges that are analyzed for eDNA, even using some of the filter feeders like this deep sea sponge as a sampler of environmental DNA. And we're exploring how deep water sessile invertebrates that filter water may act as samplers for eDNA, and comparing what we see in those tissues to water samples nearby and those taken distantly. We are marking corals for repeated visits to understand how predators or survival or recruitment are operating, or reproduction in coral polyps. We're looking at metabolic rates of some of these animals using sort of advanced deep sea in situ metabolic rate measurements, or chambers, or other sensors where we can measure oxygen coming in and out of the animals. And as far as restoration, I mentioned that these are highly vulnerable, and they've been trawled not only off Australia, but throughout areas of the world where some deep sea fisheries have started to exploit fisheries near these areas that are trawlable. Can we actually jumpstart the recovery of some of these systems by literally transplanting corals from one place to another? Very crude experiments that we started initially a few years ago to sample some deep sea corals, bring them up to the ship and plant them in small little pots like you might find in a nursery, and then take those back down to the environment and follow their survival and growth for several years to see how they do. And some of these, like the bubblegum coral you see here, they tend not to do well. Others do quite well. Even bamboo corals that we thought were the most fragile seem to start, they certainly survive and maybe even start growing, but their growth rates are quite slow, and we're still following those experiments. Along the way, we might find some really exciting new discoveries. Part of NOAA's exploration cruises that we're involved with, we found these deep sea Octopus Gardens at 3,200 meters depth, 10,000 feet below the sea, where thousands of female octopuses are brooding their young. You can see eggs here. We make temperature measurements, and it turns out that this is at the base of the Davidson Seamount, where, although it's an extinct volcano that was produced around 50 million years ago, there is still heat emanating from it, and the water around these areas where these brooders are located are far warmer than the ambient waters nearby. So that's one of the sort of side discoveries when you start looking in areas where people haven't been. Now, to bring it back to what are the problems and how are we going to move forward with this in the future, we now go out to sea, use these advanced techniques, bring back lots of data, lots of video, lots of sensor data, and that comes back to the lab and requires lots of effort to analyze those data and analyze those samples and produced real data, the understanding of what's there. And we want to move from that using some advancing techniques, both in terms of undersea robotics, where we can have advanced instrumentation that can fly near the seabed and scan the seabed, and then couple that with advances both at MBARI and elsewhere in machine learning and AI to automatically detect some of the key species that we're looking for, even make some sort of metrics of those on the fly as it's flying through over the bottom, and report data rather than just volumes of video. And those animals might be identified, counted, and even compared to environmental parameters that are measured along the way. And this sort of technique is going to be needed to scale up at in some economical way, to monitor many of the deep sea areas that we are interested in monitoring. Even some of the shallower MPAs could certainly be monitored using advanced robotics like this. And hopefully that's the direction that MBARI and collaborators will take this in the future. And I'll stop there for this, and I'm happy to discuss this in more detail. Oops.
Great, thanks, Jim. I loved how you took us from the very, very high tech to using PVC pipe to sample and plant the corals. And there's a lot that we could talk about, but right now we're going to switch gears a little bit and go to Dr. Damian Chapman, who is an associate professor at the Florida International University. And he's a microbiologist, but he is doing a lot of work with the shark fin trade. So I'm going to turn it over to Damian for more information on that. He's going to take us up, I think, more on the land side aspect of this.
Thanks very much. All right, so if you're seeing a hammerhead, this is all working. So my name's Demian Chapman. I'm a molecular ecologist from Florida International University in Miami. And this is a live scalped hammerhead shark. And like my colleagues, I like to monitor biodiversity on the water with live animals, but I'm gonna take things to the dark side and talk about tracking and monitoring dead animals, in this case, shark fins. So shark fins are a luxury product used in shark fin soup. What I'm going to be talking about today is monitoring the shark fin trade, but you could really apply the tools that I'm talking about to monitoring all kinds of marine wildlife which are threatened by illegal, unreported, and unregulated fishing. So in order to fight this IUU, we have a wide variety of laws at the national, state, national, and international level, one of which at the international level is the Convention on International Trade in Endangered Species, or CITES. And we have quite a few marine species on CITES now. And what CITES basically does is once species are listed under this treaty, you're either not allowed to trade them across international borders, or if you do so, you have to have permits that certify it's legal, sustainable, and traceable throughout the supply chain. So it's really important to figure out, are CITES and other laws actually accomplishing their goal and leading to real world outcomes on the ground in terms of improved biodiversity or reduction in biodiversity loss. And a lot of the trade in marine species is very, very much like the shark fin trade here, which is you've got a whole bunch of countries that fish for the animals, take the animals out of the water, and then the markets might be on the other side of the world. Shark fins, the market is primarily in Southeast Asia, with Hong Kong sort of being one of the big brokers of shark fins. And the trouble with this trade is it's really secretive. We don't know heck of a lot about its species composition, what kind of shark is in there, and we don't know which regions are supplying which sharks, and we don't really know, we've had now sharks, several species of sharks listed on CITES since 2013, some for even longer than that, but really commercially important sharks since 2013, and we don't necessarily know, is that keeping them out of the market? So that's where I come in. My team at FIU and with our collaborators that are based in Hong Kong, we actually go into the market and we survey shark fins. So we've been doing this since 2014, and we've sampled about 15,000 shark fins total in that period. We've been able to keep this going since 2014. And we go in the lab and we use DNA barcodes, some relatively old tech as far as molecular biology goes, which tells us what species the fins come from. And that's very useful. So this is the species composition of the fin trade that we sort of first put together from 2014 to 16. We found that there's close to 90 species of sharks, some rays and some chimeras. So it's the shark fin trade, but some of their relatives are in there too. And about 1 in 3 of the species in this trade are at risk of extinction. So that's definitely not a good thing. And if you look, the 4th most common species is actually the scalped hammerhead, the species I showed in my introductory slide. And that has been CITES listed since 2013. And we were kind of surprised to find the scalped hammerhead was so common, yet listed on CITES, because there was very little legal trade recorded for that species. So it really indicates there's a lot of illegal trade going on in the shark fin market of Hong Kong. So that leads us to the next question, was if there's a lot of illegal trade going on, where in the world should we focus our conservation efforts on? And so what we do there is we focus our DNA attention back in the lab in what we call the DNA zip code. We look at another part of the genome, which we can actually tell where in the world the shark came from that sequence. And that works because sharks are kind of like sea turtles and salmon, in that the females home back to their birthplace to breed. And as a result, you end up with DNA sequences that are sort of associated with particular regions. So we got, for example, our scalloped hammerhead shark fins in the market, and we sequenced their zip code, and we found about 60% of the fins came from the Eastern Pacific region, which is this area from the Pacific coast of Mexico all the way down to Chile. So about 60% of the fins from scalloped hammerheads in Hong Kong come from this region. And this is in a region which obviously we need to look into some conservation efforts for scalloped hammerheads. And that has particular relevance for the United States because a lot of the fins come through US ports from this region on their way to Hong Kong. So the US plays a role in this trade, even though we're not necessarily really into eating shark fin soup. So our border control personnel are really in need of tools to help them detect the trade in these species. So we are pretty interested in not just what can we do in the lab, but what can we do outside of the lab to monitor this trade. And what we've done with funding from the Paul G. Allen Family Foundation is to develop a mobile lab that we and customs agents can take to the airport, to the shipping ports, wherever shark fins and other products are coming in. This can be applied to anything with DNA, so it's not just shark fins, we use it for sharks. And what that does is it gives we actually train the border control officers to use this technology using real-time PCR, and they can actually run a test on a fin for about $1 a fin. They can do about 100 of them in 3 or 4 hours, and it doesn't require too much training, and it tells you if it's CITES listed species or not, which is very, very useful because then they can detain a shipment for a short amount of time and figure out, does it have CITES listed species in it, no matter what the condition of the fin is. So you might have heard last week, there was a bust in Hong Kong where they seized 29 tons of shark fin. So they partly do that by looking at the fins, and they partly do that by doing DNA testing. So that's a good application of this little mobile lab that we have. And we're actually going into the starting point of the supply chain. Here my student Diego Cardonosa is teaching some Peruvian border control personnel. So they're also testing the fins and other shark products with support from Oceana, Peru, to be able to detect illegal trade in CITES listed species. So I hope I've convinced you that it's great to monitor live animals and see how they recover over time with any luck. But tracking the dead ones, both by scientists and border control personnel, can also help us in that goal of restoring biodiversity. Thanks very much.
Yeah, thank you, Damian. That was fascinating. Don't often see that part of the trade and how you track it. Another example of how technology is really changing, being able to do shark conservation. So the last on our panel, we're are really thrilled to have Julie Catto here from Google, who's going to talk about some pretty interesting ways of taking some Google technology that we're pretty much all familiar with and applying it to the marine world. So Julie is a product manager at Google, and I'm going to turn it over to you to tell us more about your project.
Thank you so much, Josie, for the introduction and for inviting me to join this panel. My name is Julie. I'm a product manager in Google Research, and I'm here today to tell you a little bit about the bioacoustics effort at Google. So at a high level, the goal of our team is to leverage some of the existing AI techniques that have been developed at Google to detect animal sounds. The purpose is to help biologists better monitor and hopefully ultimately protect animal species. So almost all animals make some kind of sounds, from birds to frogs, dolphins, whales, and I was also surprised to learn that even fish can make sounds. And so hiding microphones in natural habitats is a common way to monitor animals. It's easy to collect a lot of data because devices have become cheaper and they have more and more memory. But then the tricky part is to make sense of this data, to analyze it and to draw some insights from the data. And this is something that we believe AI can be very helpful with at scale. So we're a very small team within Google Research, but we have worked on multiple different animal sounds. But today I would like to tell you about our partnership with NOAA, specifically on humpback whales, which is the beautiful animal you can see on this picture. Humpback whales are not endangered anymore, but they were in the past, and oceanographers from NOAA are actively monitoring them. So we were approached by Dr. Anne Allen from the Pacific Islands Fisheries Science Center about 3 years ago to help her on her work with humpback whales. So we learned that humpback whales produce very complex types of sounds that some say that they sing. And these are pretty difficult to detect. They can be confused with other types of sounds, and they range from low to high frequencies. So knowing where the whales are and when can enable researchers to study, for example, their migration path, or to see whether protected areas have been successful. So those are important insights for researchers at NOAA. So Dr. Anne Allen and her team since 2005 have been recording audio at the bottom of the ocean in 13 different sites in the Pacific Ocean. In total, they have accumulated the equivalent of 21 years worth of underwater audio recording, which of course is a lot, and nobody could ever listen to the whole thing. And so the question that they had is, how can we best analyze it? So what we did on the Google side is we trained a neural network that automatically identifies humpback whale calls in this very large recording. We're continuing to improve that model to recognize other species like orcas and right whales and minke whales, but we started with the humpbacks. Now we'll tell you a word about how it works. We actually leverage for that project classic image classification techniques, same that are being used to identify, for example, cats from dogs on a photograph. So the first thing we do is take the audio recording and turn it into a picture, because this is what we know how to analyze the best. So what you can see on this slide is a spectrogram, which is a visual representation of a sound. On the y-axis is the frequency of the sound, and the x-axis is time. So the sounds that are on the lower part of the spectrogram are low frequency, and on the higher part are higher frequency. And the brighter spots are the louder sounds. So spectrogram can show sounds that are too low or too high frequency for humans to hear. So they give the full picture of the sounds and are very helpful because you can annotate them. So our partners from NOAA took the spectrograms and added labels to them. So you can see, for example, here that Dr. Anne Allen told us this is a humpback whale. This is not a humpback whale, it's a ship, so that our models doesn't get confused. In general, machine learning models will work better if they have as much annotated examples as possible. In our case, we got out of the 21 years worth of audio recording, we got about 10 hours of labeled data that we used to train the algorithm. And then we worked back and forth with our partners from NOAA to evaluate how well the model was doing and to measure its performance until we reached a point where they could use it for their research. So after training the model I was telling you about, we ran it on the 21 years worth of data that I was mentioning. What you can see on this slide is just a simple visualization of the output. So it's a map of the Pacific Ocean, and it's actually an animation that shows every year some colored dots that indicate where our machine learning models detected the presence of humpback whales. So Dr. Anne Allen from NOAA is using insights from our classifier to conduct some research on the presence of humpback whales around the Hawaiian Islands. Thanks to the classifier, she made a few interesting discoveries. For example, we have the first record of seasonal humpback occurrence at Pagan Island in the Marianas Islands, which is likely to be part of a missing population of breeding humpback whales. We've also documented for the first time seasonal occurrence of humpback whales at Cross Seamount, a seamount to the west of the main Hawaiian Islands. And finally, we've also got the longest record of humpback whale in the northwestern Hawaiian Islands that shows regular seasonal humpback songs, which is exciting. So yeah, so that's all I have. Thank you so much for listening.
Thank you, Julie. That's great. Can you detect it in real time, or is this just all going and looking at historic records?
Oh yeah, that's a great question. So our partnership with NOAA was focused on passive acoustics. So we were actually detecting after the fact whether certain whales had been present in the recordings. But another project we've done more recently was with the Department of Fisheries and Ocean in Canada. And what they asked us to do is to detect real-time presence of orcas in the vicinity of their hydrophones. So actually the same technology can be applied to both applications. Yeah, and we're interested in both.
Yeah, Jim, go ahead.
Great talk, Julie. It's interesting. I just wanted to note that if you're interested in live soundscape information, if you go to the MBARI website, we have a live stream from a hydrophone in Monterey Bay, and sometimes you hear nothing, sometimes you hear a lot, but occasionally you can hear, I don't know if you can hear that now, that is the live stream, and sometimes you can hear humpbacks and other whales, and there are a series of recordings that you can play as well, just FYI for those that are interested. But thank you, Julie.
Yeah, Damian.
Yeah, I'm in charge of a pretty small data set by the standards that you just indicated, but it would be about 2 years of underwater footage from baited remote underwater video systems from a project called Global Finprint. It was a big shark and ray survey that we've done over the past few years, but one of the things I've noticed is that a lot of times in the Caribbean, when a shark arrives on the video, we hear this sound that sounds like a machine gun going off. And we think it's some sort of fish that's basically telling all the other fish, there's a shark, you got to watch out.
Interesting.
So just that your talk was very interesting, and I was wondering what it would take to get 2 years of video data combed through for this little machine gun sound.
Yeah, that's interesting. I heard from somebody, I don't know if it's true, that sharks didn't make any sounds, but it's interesting to hear that perhaps fish around them might give a hint of their presence. I think overall, for the kinds of technology we're working with, what matters the most is to have ground truth. So to be able to be certain that at a specific time we spotted a shark, and so that we can start looking for patterns of, okay, if we listen to the acoustics or look at the video.
Oh, great presentations all. I think this has been fascinating. And one of the themes that kind of came up through and through is that the need for data and this machine learning. And so I'm just sort of curious is, obviously, you know, and BARI and Google are sort of on the cutting edge of these kind of technologies. How far away are we from using these in more widespread applications? Are we still on the cutting edge, or are they really kind of becoming something that more people can use? Go ahead, Jim.
Well, Julie may want to comment, but MBARI is definitely involved in this, and I recall as a grad student, and now I have a lot of gray hair, there was a lot of interest in using machine learning to try and identify corals and coral transects from video, and nothing really came of it. But now there's been an explosion of the use of neural networks and machine learning to identify things. The crunch in those data are you need to have a test data set, a labeled data set that says this is a humpback, or this is a white shark, or a turtle, or whatever it is. You need data that shows a frame with a box around that animal saying, here's what this animal is. That training data set then can then be used to refine the methods. And MBARI has a kind of a stumbling block right now to create these labeled data sets so that they can move beyond that need and really get into refining the models. Julie, you might want to say more about this.
Or Frank also wanted to chime in. I think I'll
Yeah,
turn to
thank
Frank.
you. These were really interesting talks, and I'm very interested in the artificial intelligence. So one of the things that I think is really critical is that we all try to work together in common formats of the product, and that the product flows into common databases. And because I think that this is one of the big limitations, not only in local conservation efforts and sustainable development and any type of management, is the information is all spread all over the place in different formats and different people's computers. And so if there's an effort that we work with very closely, that's the Ocean Biodiversity Information System. They changed their name now, OBIS. And they are working with standards and standards for how you format the data to flow into the OBIS database. There's another one called GBIF. So if we can all work together to get these data streams, not just to make a product and deliver it to a one customer, but to make the data available in standard formats and open and public, that would be a huge contribution.
Great. Jen?
Yeah, I think the comment Jim made about training sets is a really interesting one, and it is going to be absolutely critical to the wide acceptance or the wide use of these sorts of AI and other technologies. And, if you take, for example, like these huge monitoring programs that we're using in California, which are a lot, for the most part, human-based. We've got humans out there not accessing the deep sea necessarily, but in some of the other habitats. We think the way to move forward is to start pairing up these technologies. So whether it's video, audio, or photographs, at the same time that you've got the experienced trained humans or the trained citizen scientists or the academic scientists or whoever they are out there doing the monitoring. And so what that will do is it'll allow us, I think, to sort of create the training sets as we're still doing the monitoring. And it will be easier, I think, than necessarily than sort of, bringing in folks to just look at these types of sets that were brought in without their experience. Does that make sense? So pairing the technologies, the high-tech stuff with the low-tech stuff, with the humans who are out there doing it anyway, and boom, you've got your monitoring going on and your training sets at the same time.
Right. Does that make sense, Julie, from your point of view at Google? And
yeah, no, absolutely. And everything, yeah, Frank, I totally agree with you. It would be the dream to have all the data source shared and accessible to others. One thing I wanted to mention is that one thing we were looking for that I couldn't find, and maybe it doesn't exist or maybe it does, we just don't know about it. But if a researcher trains a classifier to detect certain species of animals, it would be so helpful to have some kind of benchmark, a test set that multiple people can use, maybe with recordings of humpbacks from different oceans or different areas in the planet or at different seasons, so that researchers in machine learning can basically evaluate the performance of their model, but also compare it with what other people have done. And currently, I think what we've been doing is that each application we work on, we put together our own test sets to evaluate our models. And I think it would be very helpful for the research community to have more standardized test sets that everybody can use to allow for more fair comparison. So that's something that I think would be of use as well, in addition to having all the training data in one place, as you described.
Yeah. Jim, did you want to go ahead and respond to that or add to that?
Just a comment. I think that's really critical to move forward, because the community wants general tools developed so that we can apply those to different systems, the deep sea, the intertidal, soundscapes. And MBARE is involved with partnerships through a variety of institutions to try and develop those tools, both for training sets and the analytical tools, neural network tools, to look at both our soundscape data as well as some of our midwater and benthic data. And some of those, one of our engineers, Kakani Katija, is leading an effort called FathomNet to try and create some of these data sets, labeled data sets, that can be used as training data sets and make them available to everyone, literally anybody that can download them. And that's in progress now. So I'm hopeful that this is going to be a real step forward in taking what we all have on our shelves of lots of video data and being able to derive data from those, or summaries from those.
So I have a question. So we've been talking like big species. Does machine learning have applications for like phytoplankton and zooplankton as well? Are there developments in that area? Go ahead, Frank.
Yeah, I mentioned the imaging flow cytobot and the zooplankton samplers. There's several efforts to try to have open libraries of images that people can use to train their algorithms. So yeah, this is more and more. You see that in the genetics field. You see that in any type of video and imaging technologies. And I think that this idea of having a library of sounds is not new. I mean, I think that there are people who have been doing this for decades in other circles, but I think having it open for the public would be very good.
Yeah. Okay, well, I do want to ask one other question, and which is, since we're sitting here being virtual, I'm sort of curious as to what the impact of this COVID-19 pandemic is on your work and research, and how that's going to affect conservation and restoration efforts. So go ahead, Damian.
Yeah, it's a crazy time to be a scientist or any other human on this planet, I would say. But one thing that my colleagues and I are starting to see is some of the shark fin trade, for example, is slowing quite a bit. So fishermen in many countries are not going out. Or they're just unable to move product because everything's shut down. So it's it's one of these things. It's hard to know if this is going to be a pause that's good, or when this thing reopens and there's some sort of pent up demand and things just go crazy. So I think we've luckily been able to continuing monitoring Hong Kong through this, and we can do it because we have teams that are local and they don't have to travel. So we'll be very interested to see if there's any change in the species composition and sources over that period.
Yeah, thanks, Damian. That's interesting. Jen.
Yeah, I think that, as Damian said, this has been super challenging at a lot of different levels for scientists. From sort of the California perspective of the field ecologist, obviously, people are not able to access the field right now to continue monitoring, for example, of the marine protected areas. But I think if this panel has shown one thing, it's that I think this emphasis on tech and having things out in the ocean that are monitoring without the people associated, to the extent that that's happening, is one way to potentially fill those gaps, remote sensing and so forth. So there are still a lot of data streams coming in that are not reliant on having eyes in the field or boots on the deck or a boat on the water. To the extent that continues, even those data streams will eventually fold if they're not maintained. But that's what we're thinking about in California, is how to fill these gaps when the people are not out in or on the water.
Right. Anybody? Yeah, go ahead, Jim.
Let's make one more comment about that, because I mean, everything that Damian and Jen said is just right on the money, and we can't run our ships right now. We're trying to figure out how will we do that once we get back out there. There are time series observations, just like for the MPAs, that we'd like to get out and get back to work on. But on the other hand, maybe this is a bit of an opportunity for figuring out what goes on when we stop running ships all over the place. The soundscape work is one where I think some researchers, both through Mbari and some collaborators, I've forgotten who, are, because I'm not in that realm, are looking for a rapid response grant to try and assess, are there changes in either mammal distributions around the hydrophone related to the reduction in ship traffic and things like that. So on one hand, it's really terrible, but is this an opportunity for some research that is unprecedented? Is, I guess, a question I'd pose.
A real-time experiment, you know, can we monitor it while it's happening? Yeah, Frank, I'm going to turn to you for the last comment.
Yeah, I think that this is a very important comment. One of the things that is very important about biodiversity is that it is a source for pharmaceuticals, for materials, for different types of food and protein that we all depend on. And so it is really important to find ways to continue our exploration of how and what lives out there and how it is beneficial, not only for the environment, how do we conserve it, but because it is beneficial to us.
Yeah. Well, that's a perfect note to end on, Frank. And I want to thank all of you. This panel was fascinating. It's just exciting to hear the range of different work being done on marine conservation and sort of the promise of new technology, particularly this machine learning, and how diverse and vast its applications can be. And I think it's going to be a real transformative technology for us in doing conservation work. So I want to thank you all, and we'll be in touch. Thanks.